even just saying a yeast genome, yeast tend to mutate quickly. So there's never really a finished yeast genome. So assembly is an imperfect process, especially for a complex genome like human, but assembly has been improved over time. And in fact, one of the biggest improvements happened just in the last few years when we developed things like PacBio and nanopore sequencing. So in the genome, there's a lot of repetitive regions, and there's also a lot of these insertions. There's what are called line elements, sign elements, allorepeats, and these are actually bits and pieces of old viruses or transposable elements or all these mobile genomic elements that can jump around the genome. And the problem with them is, for example, allorepeats, you can pretty much find everywhere in the genome. You'll find them on every chromosome, usually in a few different places. Actually, and lines and signs, which are other repetitive elements, you'll find them in a lot of different places all over the genome. So the problem with these is if you just do a short read, you'll know that you're reading, for example, a line element or a sign element or an allorepeat, but if you can't read across it, you don't necessarily know where you're reading. So getting the latest reference is usually a good idea. There's a lot more things that are properly mapped and that we know where they are, and there's a lot better identification of some other difficult situations. So another thing that happens in genomes, including human, are pseudogenes and orthologs, which is where you can have multiple copies of the same gene in different locations, or multiple copies of something that maybe used to be that gene and is inactivated in different locations. And those pseudogenes also tend to really mess people up, especially because the pseudogene looks like the real gene, typically with just a couple of polymorphisms or a couple of small base changes or maybe an insertion or deletion. And if you're unaware that there's a pseudogene with that particular mutation, you'll think that you're looking at the real gene with the mutation because that's where things will align. So human references, at least canonical human references, are available from two different places. The Genome Reference Consortium and University of California, Santa Cruz. Both organizations have slightly different ways of identifying chromosomes. So if you get a genome from one from GRC, I believe they just call chromosome 1 the number 1, and they call the mitochondrial genome, I think, MT. The UCSC genome calls chromosome 1 CHR1, chromosome 2 is CHR2, the mitochondrial genome is CHRM. And the problem is if you try and use these if you try and align to a UCSC genome and then you run through a program using the GRC genome as reference, the two programs will think you're trying to run on two different genomes, and they will basically crash and tell you you made a mistake. The other thing is the current version of the human reference genome is 38. So typically a GRC genome is referred to as GRC and then H for human. So GRC H38 is a current GRC genome. For UCSC, they just call it HG. So UCSC's current version is HG38. This sounds like it's kind of pointless because I'm just saying they're using the same number, but actually this is a big deal. So the previous version of the human genome from GRC was GRC H37. UCSC had a different way of counting major and minor draft revisions. So the last version of the UCSC human genome was HG19. So for UCSC, it jumped from HG19 to HG38 so that UCSC and GRC could actually be in agreement with each other on version numbers. Otherwise, it was kind of a nightmare to deal with. And another thing I want to talk about is decoys. And decoys are actually very important. They're very useful for human genome. So I'm going to ask a question out there to the Zoom chat. If I were to draw a sample of blood on myself and extract the total DNA from it, what species would I see? And yeah, feel free to give the obvious answer on that. What species would I see? Yeah, I'd see human. And that's kind of the obvious answer. Would I see 100% human DNA in there? Who out there thinks I'd see 100% human DNA? All right, Marco says no. Anybody out there have an idea of what else I might see besides human? All right, somebody says I'll see bacterial DNA. Yeah, that's a distinct possibility. There's always been this idea that the human bloodstream is completely sterile and has no microbiome, and I think that idea has been pretty thoroughly shot down. In addition to that, blood has to be drawn through the skin, and there's always been a well-known skin microbiome. And sequencing is sensitive enough that I'm pretty much guaranteed that I'll pick up at least a little bit of material from wherever the needle passed through, including my skin. Anybody in this crowd interested in virology or know much virology? What else could I see? Okay, so viral transposons, that's a possibility. Anybody out there ever heard of Epstein-Barr virus or cytomegalovirus? So there's roughly about the same number of people walking. I think there's probably more people walking around with Epstein-Barr virus in their blood than not, or at least Epstein-Barr virus sequence. Because when it causes mono, and once you get it, it sometimes hangs around in white blood cells for a very, very long time. There's also cytomegalovirus, which unless you're otherwise sick, cytomegalovirus doesn't really do anything to you. It just kind of hangs around, but if you do get sick, it does some pretty nasty things. So cytomegalovirus, I think in the US, about a third of people are walking around with that. And I think in Europe, it's even slightly higher, it might even be close to 50% of people are walking around with cytomegalovirus. So there's gonna be things in there that are definitely not human. The decoy genome addresses that issue. So basically the idea of a decoy genome is you have all of the typical human chromosomes in there. You also have all of the human sequences that are kind of considered unalignable. So there's a lot of segments that we know are human, but we don't necessarily know where they align properly, or we just can't align them properly. So another example is ribosomal RNA genes. Those are all over the place in the genome as well. But the decoy genome goes a step further, and it takes a lot of sequences that you can expect to find if you're dealing with a human, and it gives them something to align to. So that's actually a good thing, because you don't have to worry about your aligner struggling with that. The aligner will actually have a place to put it, and it'll be a proper location. And then once it's done, you can just kind of get rid of the decoy stuff if you're not interested in it. All right, so dealing with alignment, actually gonna jump over this one for a moment. So when you're dealing with alignment, and actually let's get everybody onto the Hoffman cluster. So I'm going to open up another slide really quickly. All right, everybody should be seeing a slide with instructions on how to connect. Oh, oops, let me actually fix that. Sorry, PowerPoint likes to capitalize things even when it shouldn't. Okay, so we're gonna skip. So you'll remember this slide from yesterday. It's just kind of the instructions on there. The stuff that's grayed out we don't need to do today because we did yesterday, but you do need to get a new interactive session going. So that command is going to be qrsh-l, and then h_data equals hg equals 8g. So we need, apparently due to some policy changes on the Hoffman cluster, we need to request a specific amount of RAM. So in this case, we're going to ask for 8. That'll give us more than enough to do what we're doing today. And yeah, the decoy genomes, may or may not deal with pseudogenes and orthologs. So the decoy genome, previously the decoy genome would definitely want to have pseudogenes and orthologs in there as just decoys. A lot of the pseudogenes and orthologs, thanks to better sequencing, we now know where in the genome they are, and they've just been basically placed there. That being said, if there is something that's a pseudogene or an ortholog that we still haven't placed in the genome, but we know there is a pseudogene for something, it would definitely go into a decoy genome as basically a decoy to make sure that it does, because pseudogenes and orthologs are especially nasty because if you don't know, if they're not documented in the reference genome, then they have a bad habit of being aligned to the real gene and causing a lot of confusion when looking for mutations. All right, so I'll give everybody a minute or two to get logged in and then get their interactive session going. All right, quick show of hands, who's gotten their interactive session started? Okay, looks like everybody's got their interactive session started. All right, once you got your interactive session started, you'll want to CD into, back into the working folder that we're, oh, yes, do pay attention to what Natalie said. You can't have a space in between your equal sign and your, you can't have a space with the H data equals 8G. Yeah, so anyway, you'll want to CD into the working folder. So it'll just be CD and then $scratch. So I put the command for that in there in case anybody needs it, but that's how you get into your working folder for the class. All right, so on the Hoffman cluster, there's something really cool called module or module load. And module load actually makes programs available to us. So it'll load all the dependencies and it'll actually prepare our instance to run whatever we need. So the first command we're going to run, now that we have our interactive sessions going and we're in our workshop 2_data folder, is we're going to run module load. So it's going to be module load BWA because we're going to run an aligner called BWA or Burroughs-Wheeler aligner. Once you type module load BWA, if you hit enter and don't get any error messages back, then it's working as it's supposed to. Next thing we're going to do is hit LS. And LS, we're just going to look and see what we have. So we'll see the stuff we have from yesterday. And we'll notice a couple things that are going to be useful for us when it comes to alignment. So particularly, we realize that we have a reference here, which is probably our reference genome. So let's CD into the reference folder and do LS, and you'll see that you have something called chr14.fa. Anybody want to take a guess as to what that is? Yes, chromosome 14 reference genome. That is human chromosome 14. Anybody have an idea why we're only working with chromosome 14 today and not the entire human genome? Yeah, save time. What we're going to do with chromosome 14 is going to take only a couple minutes. Doing the operation with the entire human genome takes a lot of data, and it takes an hour or two. All right, so we're going to do cd ... to get out of the reference folder, and we're going to type in BWA. And BWA will run BWA, but we're obviously running it without any specific command. So BWA is probably just going to show us either an error message or a little splash screen telling us how to use it. And in fact, you should see something that looks just like this, giving you some basic BWA instructions. So is anybody out there not able to see this little splash screen giving you BWA instructions? All right, so the next thing we're gonna do is we're gonna try and run BWA since we have a reference genome. Let's see, I have a question coming in. Under BWA command, there is an index option. How does this work? That is an excellent question. I'm not gonna answer that right now because we're actually gonna learn about it in depth in about 5 minutes. But I like your thinking. It's actually about to become very important for us. So the command we're gonna run is BWA. And BWA has to have a mode it's running in, so it's going to be ALN for alignment. We're going to pass it the reference genome. And you notice this one doesn't have any -O's or -F's. So that means the arguments are all positional. Or in other words, all the information you're giving it, everything has to go in a specific position. So BWA always knows that the first thing you're going to give it is the mode it's running in. the next thing you'll give it is the reference genome. And then after that, you're going to give it a file that it's going to be analyzing. And does anybody remember what this greater than symbol actually does on the command line? It specifies output, yes. So a lot of people who work on the command line call it a T, spelled T-E-E. And they'll frequently say, I'm going to take the output of this and T it to this file here. Oh, one thing of note, I actually have a little typo in this one. So the file we're generating is not a FASTQ. So it should be 3 BWA, it should be all this, but instead of .fastq, it should be .sai. Now it turns out the command line doesn't really care what you call a file, but the file we're generating is actually what's called a SCI file, or a sequence alignment index file. So do call it a SCI file, although in this case it's not going to be as important because this command is actually going to fail anyway. So a lot of people are going to see this message down here saying failed to locate the index. It kind of tends to blend in with what looks like valid output up here. And this is kind of my main annoyance with BWA. I absolutely love the program. I think it's, you know, it was basically the program that saw us going from being able to read DNA faster than we could align it to a genome to being aligned DNA to where we could align DNA to a genome faster than we could read it. So anyway, BWA, as much as I like it, it has bad error messages. So this one failed to locate the index. This one's a little bit more informative, but I've actually seen error messages from BWA saying things like unable to perform Psi magic, which is completely uninformative. And it turns out that's the error message you get if your Psi file is somehow corrupted or blank. So anyway, so here it says failed to locate the index. So, oh yeah, and just a reminder, this T, or you know, this specifying the output, there's no safety in it. This is a really good way to accidentally overwrite files that you don't want to overwrite. So yeah, we got a couple options. We can get mad and destroy our computers, or we can just read the error messages. So again, the error messages in BWA tend not to be super informative and not always very clear that there was an error. But in this case, there was an error. So it says failed to locate the index. And I know that Shuya Wang mentioned this a few minutes ago about the index option, and now the index option is going to become important to us. So if we look even in just this little set of instructions, it says, to use BWA, you first need to index the genome with BWA index. So actually, I'm going to skip that one for now. All right, so the command we're going to run is BWA index, and we're going to just pass it the reference /chr14.fa. So in this case, we're just going to run off of chr14.fa. When you hit enter on this command, it's going to take a couple minutes. And as you see here, when I ran it previously, it took about 5 minutes to run on chromosome 14. So once you have this up and running, let's see, it's 9:47 right now. I think we'll pick back up again at We'll pick back up again at 11. That'll give us time for anybody who's having issues with it to get them resolved, and it'll give us about 5 minutes to run. Although from what I hear, the newer I made this slide when Hoffman had an older storage system. From what I hear, the newer storage system is much better performing, and it actually finishes in about 2/3 the time. But I'll give everybody till 11 o'clock. We'll get this sorted out, and I'll be around here in case people have questions on what's going on here, or if they're having trouble getting it to work. Oh yeah, sorry, I mean 10 o'clock. Yeah, everybody be back at 10 o'clock, sorry. All right, I think everyone should be heading back. I'll give everybody a minute or two to get back to their computers if they're not. All right, I got a quick question about trimming, and I think we'll begin. So Mo Ishihara has a question of, oh. All right. Mo Ishihara has a question about trimming. So actually I'm gonna, Mo, would you be able to ask that in the main chat so that it goes out to everyone? I generally like people to see, oh, okay, there we go, thanks. Okay, so the question is adapter trimming also a smart trimming? And the answer to that is yes. Adapter trimming actually has to generally be smart trimming. You can't really do hard trimming of adapters unless you know that all your inserts are exactly the same size and you know exactly how many bases you read into your adapter. So as a general rule, adapter trimming usually has to be actually very smart because it has to not only figure out just the quality score, but it also has to figure out, based on the qualities and based on the sequence, what's the probability that the sequence at the end of your read is actually adapter. That being said, because adapter trimming generally can only happen a very limited number of ways, people don't usually classify adapter trimming into smart or hard trimming. They just usually say that it's adapter removal. So does that answer your question? Cool. Okay, so what you saw was that we did the BWA index. Burroughs-Wheeler indexing is actually something that's really computationally complicated. It's sorting all the bases by their context, and it takes a long time. The good news is once you've indexed your reference genome, it's indexed and you don't have to do it again. And it turns out the reason that you do this is you kind of front-end load your alignment operation. So you index once, and then every time after that, you can do very fast alignments. So we're going to do our alignment and mapping now. And the alignment and mapping, that's going to take place in basically 3 steps. So there's doing our, we still have to do our sci files, and the sci files are not actually alignment. The sci files are kind of BWA generating a cheat sheet for itself, deciding where each of the reads might go. So the initial step is going to be this one right here. Remember, you want to call the file 3bwa human 001 pe1.sci, but it's going to be this command. So it's going to be what we tried to do before, BWA align mode. We'll give it the reference genome, which is now indexed, so it should work. We'll give it our FASTQ file, and we're going to tee it to the sci file. Another thing you'll notice, who out there is, just raise your hand really quickly, who out there is familiar with the idea of doing the up arrow on the command prompt to get back your previous command? Okay, excellent. So if you haven't done it before, this is actually a really good time to get familiar with that. It's going to make your life so much easier. After you run this first command, hit the up arrow, and you'll see that you have you'll see that you have your first command back. Because the only difference between the first command and the second command, just like previously, is the PE1s. So this PE1 changes to a PE2, and this PE1 changes to a PE2. Everything else is identical. So all you have to do is change a pair of 1s to a pair of 2s. And then just hit enter, and it'll run that command. So these two steps are each going to take just a couple seconds. Yeah, when I ran it before, it took about 45 seconds each. I think now it runs even faster. So I have a question. Can I explain what indexing is again? Yeah, so indexing is taking all of the bases and sorting them by their context. I intentionally don't get into detail into indexing right now because the actual thing that's going on behind the scenes in indexing is very computationally intense. And I have a question from Soyun Kim. Missed it. How do I change PE1 to PE2? So all you do is you hit the up arrow once that first command is done, and you'll get back your original first command. Then you can use the arrow keys to just to go back to the PE1s and change them to PE2s, just like editing most other text. Okay, and that looks like you got it. Yeah, it's so much easier than rewriting the whole command. All right, and somebody's having a problem. Failed to open the PE2 FASTQ file. Okay, so if you can't find that PE2 FASTQ file, the first thing to do is type ls and see what files are in the folder. And see if you have something that looks like it should be that PE2 FASTQ file, maybe with a typo. And if it's the PE2 FASTQ file with a typo, you can either change the file name or you can enter the file name with a typo. So for Madeline Sholto, when you hit ls, do you see something that looks like it should be the right file? Or is the right file missing? Okay, yeah, and like Marco says, it's always a good idea to use tab autocomplete. So the nice thing about tab autocomplete is if you have a typo, tab autocomplete will just complete it with the typo included. And that will actually work. All right, so you're saying that it's there. Can you copy and paste? Yeah, copy and paste. So it looks like you copy and pasted the error message. Can you also copy and paste the file name that looks like it should be? So when you hit ls, you should see a file name that looks like the PE2. Okay, so that was actually copied and pasted from the terminal. Okay, try just doing it using tab autocomplete and see if that fixes the problem. There might be something very subtle there that I'm just not seeing as a difference, or there may have been a typo somewhere. Ah, so Marco saw something. Although, if both versions of the file, both the one you're calling and the one that's stored, have the dot, then it's fine. Okay, looks like I got it. So if you want to change a file name, the command to change a file name is actually the same as a move command. So does anybody remember what the Linux command is to move a file? Yeah, MV. So it's MV from the source to the destination. And in this case, we're actually not moving it between directories. We're just moving it from one file to another. So it would be MV from the old file name to the new file name. And that functionally changes the file name for you. Okay, so I think we can get going again. All right, so as I mentioned before, these SCI files are not human readable, and they're not actually the alignment. They're just sort of a cheat sheet. The command we're going to run is BWA and then SAMPE. So this is actually what's going to generate the SAM file, which is our desired output. So SAM paired end mode. We're going to give it our reference. Yes, we'll give it our chromosome 14 reference. We're going to give it our forward and our reverse SCI files that we just generated. We're also going to give it our forward and our reverse FASTQs that we just generated. And then we're going to output to a file that we call align-pairedend.sam or align-pe.sam. And that'll need to run for a minute or two as well. Okay, so I think everybody should have gotten that to run. You probably got a thing saying unexpected end of file. Did people see an error message saying unexpected end of file? Okay, everybody's saying yes. All right, so again, we have options. We can get mad, or we can double check what we aligned with. So keep in mind that we made these sci files using our scythe and sickle process files, so the ones that start with the number 2. So all we actually have to do is go back and just change those original FASTQs to the sickle processed files, and then it'll work fine. I intentionally walk people into the error because this is a common error that I've seen made, where people will generate a sci file and then accidentally go back to the original for the alignment when they generated the sci off something processed. And unfortunately, BWA again doesn't give good error messages, so that unexpected end of file doesn't really tell you as much as maybe saying that your sci files and your FASTQ files don't line up. Did you use the same files? So give that a run, and I'll give everybody a minute or two to do that. All right, did people get the latest one to run successfully? Okay, cool. I'm seeing a lot of yeses. Sorry, give me 1 second. My computer is acting weird. Sorry, give me just 1 second. My computer is acting weird and PowerPoint stopped responding. Okay, there we go. I think it came back. All right, did everybody see the slide change? Okay, cool. Not sure why it was doing that, but it was acting weird. Anyway, okay, so where are we right now on the pipeline? So we've aligned our raw reads to the genome, and that actually oh, we have a question, what was the command we needed to run? So I'll go back up a slide. So this is a command you need to run, and again, it's saying BWA, run it in SAM paired end mode, and it's going to look at the reference. So it always needs a reference, and it's going to be taking our forward and reverse sci files and our forward and reverse FASTQ files. And and actually the command's up here, but it's the same thing, but it's going to be taking our forward and reverse sci files and our forward and reverse FASTQ files, and it's going to be keying the output to aln-pe.sam. So for Madeline, does that, the sci files are kind of like cheat sheets for BWA to do its alignment. So it's going to align the paired ends individually first and come up with a list of places where they might go. And then once it does the sci process for both of them, the SAM-PE process is taking the two sci files as well as the sequences themselves and figuring out what the best explanation is using the paired end data to align it. Does that answer your question? Cool. All right, so let's have a look at what the and what we get back. So if you were to run head on that file, so if you got the unexpected end of file, that means that you were probably still using the if you got unexpected end of file, chances are that you were using the original FASTQs that were unprocessed when you should be using the sickle processed FASTQ, so the ones that start with 2. It's probably not that dot versus underscore issue, because the dot versus underscore would give you an error saying the file wasn't found. In this case, the error was unexpected end of file, which is typically what you get if you're using sci files and if you're using sci files and FASTQ files that are not aligned to one another. So it's probably not the dot versus underscore. It's most likely mismatched files. Yes, okay. So the command that Jennifer Forsyth just posted, that would be likely to give you the unexpected end of file error. You would need to change both of those human 001s to 3_sick. It would basically be giving it, or not 2_sick. It would be using the processed FASTQ files. Yes, what was just posted. All right, so once you've gotten that command to work, you've actually gotten your first, you've actually run your first alignment. So what do you see when you do an alignment? You'll get back a file that looks sort of like this. So this is called a SAM file. Who out there has worked with a SAM file? Just hit the raise hand button, a few people. Who out there is more familiar with a BAM file? Okay, a lot of people out there more familiar with BAM files. Okay, so it turns out the difference between a SAM file and a BAM file is actually pretty minuscule. The real difference between a SAM file and a BAM file, in fact, is just that the BAM file is binary and compressed. Oh yeah, you won't find this file in there. This was actually an old name, but you'll still find the same data. don't worry about this file name. This is actually just a previous file naming thing. But anyway, so what you'll see if you look in a, in fact, what you're going to do is, if you want to see the file, you can type head, and it'll be a different command for you. It'll be head and then aln-pe.sam, because that's the SAM file you just created. This is actually just an example of a SAM file. So in a SAM file, you're going to have headers, and the SAM file headers are going to tell you things like your sequences. So it'll tell you this was made with a reference to chromosome 14. It'll tell you the program that was used to run it, and it'll tell you the program used was BWA. It'll tell you the version number, and it'll even tell you the exact command somebody ran to do it. Yes, if you align to the entire genome, to one of the sort of canonical human genome drafts, this header portion would be like about 200 lines long, because there's a ton of stuff there. So yeah, the header section in a whole genome alignment is going to be incredibly long. It's going to list every decoy portion. If you use a decoy genome, it's going to list all the sort of unalignable portions of the genome that still have sequence entered in there, and it'll list all the canonical chromosomes. So you'd wind up with headers that are about 200-ish lines long or more. So anyway, these are the actual data lines. And the data lines have, as you see here, a ton of information on them, including sequence and quality score. And let's go over what's actually in them. So just looking at the first line, we see that we have a query name. So this is a unique identifier for each read, but it'll be shared between two reads from the same insert. So that basically means if you have a paired end file, you'll see the PE1 and PE2 both have the same query name. This flag score is very important. So a flag score is a representation of a bunch of true-false values that describe your alignment. So the way that this works is, anybody who's familiar with kind of how binary numbers are formed, this will make sense to you, but you create a number with a bunch of true falses in binary. But the most important thing to remember about your flag value is really that it's a representation of a bunch of true false characteristics about your alignment. So for example, the first thing is just read is paired or read is not paired. So if read is paired, it gets +1. If read is not paired, it gets +0. If the read is mapped in a proper pair, and that means the right orientation and approximately the right distance, it'll get +2, or again, if this is false, it'll get +0. 4 indicates the read was unmapped. 0 if the read was mapped. And another one would be read is first in pair, that would be 64. So as an example, 66 would be read is mapped in a proper pair, and the read is first in pair. It's kind of important to note, though, that 66 is actually an impossible flag value to see. You will never see a flag value of 66 for the simple reason that if the read is the first in pair and the read is mapped in a proper pair, which gets you 66, the read must be paired, which will add 1 to it. So 67 is a possibility. 66 is not a possible flag score. The good news is, if you want to figure out what flag scores mean, you don't have to do all this by hand. There's some pages online that'll actually help you explain flag scores. So you can punch in a number and get back check marks for all the things that it indicates, or you can check off things and it'll give you a number that comes out to that. So these are all the values associated with all the possible things in the flag score. So how are these numbers formed? It's just like when you count in decimal. So when you count with decimal, which is what people are used to, each digit is worth 10 to some power. So the first digit is worth 10 to the, so the first digit of a number is worth 10 to the 0 power, the next digit is worth 10 to the first, then 10 to the second, and so on. With binary numbers, it's just each digit is worth 2 to some power. So the first digit is worth 2 to the 0 power, which means it's the ones. The second digit is worth 2 to the first power, so it's worth either 2 or 0. The third digit is 2 to the second, so 4, then 2 to the eighth, which, or 2 to the third, which is 8, and so on. And just by convention of computer science, 1 means true and 0 means false. So flag tells you a lot about mapping. You can use flag scores to filter out a lot of things. So you can filter out unaligned reads. BAM files include unmapped reads. And it says here you want to remove these, but I found lately that people tend not to remove them. People tend to kind of dumpster dive them and see if they can figure out where they came from. A lot of people are now looking for kind of the more unusual signals, and a lot of those you'll find in unmapped. So the next thing you'll see is CHR14. So that's just the reference sequence name, or usually the chromosome it mapped to, or the contig it mapped to. You'll get a position, and you'll get a mapping quality score. And it turns out this is just another Phred score. So as I said before, the Phred score rule of thumb, a Phred score of 0 is 0% confidence, a Phred score of 10 is 90%, 20 is 99%, or a 1 in 100 chance of error. A Phred score of 30 is a 0.1% chance, Phred score of 40, 0.01. So a Phred score of 60 indicates a 1 in a million, about a 1 in a million confidence, or 1 in a million chance of error, so fairly high confidence. You'll also get a cigar string, and the cigar string is a little bit unusual, but it kind of makes sense once you see how it's coded. So the cigar string is just a combination of some number followed by some letter. And the most common cigar value, the most common cigar operators you'll see are match, which is M. And keep in mind that match can actually indicate still a base mismatch, but it's not an insertion or a deletion. An insertion is a base in your read that doesn't exist in your reference, and a deletion is a base that doesn't exist in your read but does exist in your reference. And then 9M, so 9 more matches. So basically the way this works is 80 bases aligned, and there may be some base mismatches in here, but they still see the same number of bases. There's 3 bases that appear to be inserted, and then there's 9 more bases. So the next values you'll see, mate reference position, this is where the mate mapped to. And if it's an equal sign, that just means it mapped to the same chromosome. Then the position the mate mapped to, and that'll be a number. You'll see your template length. You'll see your bases. So this is your actual sequence here, and your qualities for the bases. And then down here, you're going to have a bunch of these predefined tags. So these predefined tags are just describing different things that we're not going to worry about for this course. Okay, and that's actually everything I'm going to cover on this part. And give me a moment because I'm going to do a screen share of a different section. So I have a slightly different ending for this class than I used to.
Michael, I have a question.
Uh-huh.
You're gonna be ending class pretty soon?
It should be, yeah.
Like at 11 o'clock?
Yep.
All right, cool. Thank you. Taking attendance.
No problem. All right, there it is. Let me just pull that up. And let me get my chat window back. Okay, I have a question. What is the deadline to finish the quiz if you're taking this course for credit? I don't know if there's an officially set deadline. I'd recommend obviously doing it sooner rather than later so you remember the material. I'd actually say probably try and get it done today or tomorrow. The quiz is only 10 questions, so it should be fairly quick. But yeah, try and get it done today or tomorrow. I think probably I probably want to cut it off. I'd probably want to cut it off on Sunday if I had to issue a cutoff date, just because at some point I'm going to stop checking to see. At some point I'm going to stop checking to see if people do it. All right, and is this portion of the slides posted somewhere? So all of these are available online. There's a Google Drive where I'm putting recordings of the course. So I think a link was already sent out with the Google Drive, and I basically add more material to that as I generate it. All right, so how your aligner works. There's been a lot of questions when people get sort of what I call aligner garbage or aligners behaving badly. So I want to talk a little bit about aligner logic. So this is what's called brute force alignment. And if I just told people, here's a bunch of short sequences, and then here's a really long genomic sequence, and I want you to figure out where in the really long genomic sequence the short sequences came from. This is the approach that pretty much everybody would take initially, which is I'm going to take the short sequence, and I'm going to kind of slide it along the longer sequence, and I'm going to see how it matches at different locations. So for example, here we see it matches up okay in a few places. We move over one, we see we get a couple more matches. And in fact, both times we get, so the first time we get 5, the second time we also get 5. So probably 5 is just a number of random, about 5 is a number of random matches we can expect from 2 sequences with one of them being this length. A little bit better here. We're up to 7 matches, but we're still well in the range of, hey, this could just be random. Here, okay, it's looking a little bit better, but still quite possibly just random. Eventually we get to right here where the sequence really seems to align perfectly. But we still have to actually go, we would still actually have to keep moving the sequence across to make sure that it doesn't match anywhere else just as well. So that's a lot of operations when you think about how computers doing it. Essentially, assuming we have 50 base pair reads and we have a 3.3 billion base pair genome, which is about the length of the human genome, and we have 50 million reads from an experiment or a flow cell. I don't know how to say this number, but it's a big one. So the approximate computation time this would take is about 130 years on a single core computer. Now, to be fair, if you use a dual core computer, this would only this would still take a really long time. If you used a 4-core computer, this would still take a really long time. And basically, there's not enough cores in a computer to make this reasonable. So here's what the real challenge is that we're dealing with. How do we deal with insertions and deletions? Because I didn't even talk about that. And how do we deal with, how do we make these alignments finish before we have grandchildren? So I'm going to talk about indels first. When I say indels, I mean insertions and deletions. I'm going to talk about these first because they're the simplest way to understand. So here I took that alignment and I just generated a little deletion in it. And now it doesn't align well at all. So the way we deal with this is called seed-based alignment. And for seed-based alignment, you're actually just aligning pieces of a read rather than the whole read itself. So for example, this piece aligns here very well. This piece aligns here kind of poorly. It also aligns here, but kind of poorly. It also aligns here equally well. This piece, we ultimately get a perfect match right here. And by knowing this, we can say, we know this piece goes here, we know this piece goes here. We have this piece in the middle. Let's see if we can explain it with some kind of a simple insertion or deletion type thing. And it turns out that we can explain this very well with a little deletion model. So another thing is to understand how your aligner gets to be confident in the alignment scores it gives. So this one's kind of a gimme. If you have the same sequence and it can either be aligned here in the genome or at this other location in the genome, but at this location it has two bases missing, and at this location it has a perfect match, obviously your aligner is going to be much more confident in this one. In this scenario, we have an alignment here with 2 mismatches, and the next best alignment is here with 3 mismatches, versus a scenario where, again, your best alignment is with 2 mismatches, and your next best alignment here is with 1, 2, 3, 4, 5 mismatches. So in this case, your aligner is going to be much more confident in this scenario because the alignment it calls is much stronger than the next best alignment, versus here where you have 2 errors versus 3 errors, and it's going to look at this and say this alignment and this alignment are almost equal in strength or in confidence. A little bit more complex is a situation where you have a little deletion versus a couple of mismatches. And this one's going to depend a lot on your error model. Typically, deletions give you a big hit to your confidence, because if you can put enough insertions and deletions in a read, you can align it anywhere. So there's a very big penalty for opening up an insertion or a deletion in a read versus just a couple of mismatches. And let's see, I think this is pretty much where I'm going to stop. Or this is kind of the end of the main class. If you want to understand what's happening behind a Burroughs-Wheeler alignment, I'll show you kind of really quickly to give you an idea what's going on. So a fictional store called Mama Mab's Panama Bananas, and I call it this for the sole reason that it has a lot of repeating letter patterns, and it doesn't use a lot of different letters. So if I wanted to align letters to the sequence, right? If I wanted to align letters to the sequence, for example, AMA, I can align AMA there, I can align AMA there, actually overlapping. I can align AMA there. For Anna, I can align there, or there, or there. So one of the ways of dealing with this is something called a suffix tree. And all I'm doing is I'm taking letters and I'm building a tree structure out of them. So bananas can go here, just every letter beginning a sequence. You can see if I take off the B, now I build up over here. If I take off the A, over here. Then I actually build up over here because it turns out I have the same sequence. I build up over here because, again, I have a repeated sequence. I now have another branch over here, and then S winds up over here because S is really indicating only one position. This is called a suffix trie. To turn it into a suffix tree, I actually just have to prune it. And as you probably notice, the moment I have a B, there's only one way I can go. If I have an A, there's a couple different ways I can go. If I have an AN, there's still a couple ways I can go. So that gets a little bit more tricky. So I'm just going to prune off all these excess portions, and this is called a suffix tree. And these are good, but they're very complicated to store in memory. But they're actually way more efficient, because now trying to align something, I'm not just sliding along a sequence. I can actually just follow this tree down now that I've built it. So for example, if I get NANA, I just go to N, and then A is going to always be the next letter, and then N. And once I see this N, I already know where I am. I'm at index 2. So 0, 1, 2. And Burroughs-Wheeler is using a fancier version of this that takes up less computer memory and goes a little bit faster. But essentially what Burroughs-Wheeler is doing is it's giving you kind of a hacked version of this that runs a lot more efficiently. So I know somebody had asked what's going on when you do Burroughs-Wheeler index. And essentially you're building up a data structure that is, it looks on its face different from this, but really it's building up a suffix tree. So Burroughs-Wheeler, you're going to append on a character that sorts to the beginning. So this dollar sign actually sorts as before the letter A. And you're going to do what's called a rotation matrix. And you can kind of see if you just follow the dollar sign how we're just rotating everything around. We're going to sort the rotation matrix. And now that we have the sorted rotation matrix, we're going to take the last column. And that last column is going to give us this. And it turns out a very interesting property of doing this is you get a lot of letters that repeat multiple times. And this can be compressed very efficiently. For example, instead of saying A, you now just have A5, and then dollar sign and A2, and that's called run length compression. It compresses very efficiently and it decompresses very quickly. So I think that's as far as I'm going to go in here. I don't think I have any hardcore computer science people. But I will answer any questions anybody might have. Otherwise, everyone's free to go. Please remember to do the quiz that Eloy sends out. And as I said, I'll stick around for a few minutes and answer questions. So you can definitely use IGV to look at your sequences. I actually helped debug part of that program. I typically don't go over IGV in this course because IGV runs in Java, and a lot of people were discovering, especially if they have MedNet computers, that the Department of Medicine likes to not run stuff in Java. So I was having a lot of students having security issues with their computers when I was trying to do this course. So I started leaving off IGV. But IGV is actually fairly intuitive, especially if you just want to look at some reads. So definitely IGV is a good option. Don't worry about silly questions. It's an intro class, so I expect a lot of very basic questions. And I have a request for the Google Drive link. Let me see if I have that up. I do. So I have a question. What file format do we have to load for IGV for the reference genome? So it generally wants reference genomes in, let's see, yeah, it generally wants your reference genome in FASTA format, although there's a lot of reference genomes it'll pull from online. And then it's, so previously ran into issues getting that to work. Do you remember what issues you were having getting it to work? Okay, you may have trouble loading a .fa.gz file because that's a gzipped FASTA. You may have to decompress the FASTA file first. So you'll just need something to do to unzip the gzip file. Yes, you can unzip on Hoffman too, for sure. There should also be some programs in Windows that can unzip, that can open up a gzip file. You may have to do some looking to find them. But I think, for example, a program like 7-zip does a good job of that on a Windows machine. Are there any additional questions? All right, if there's no additional questions, I will end the meeting. Everyone out there, you're free to add me on LinkedIn. I'm usually pretty easy to find. If you have any questions, feel free to email me, or if you need help with a sequencing study down the line, I'm definitely able to give some advice. Otherwise, I will see everyone around. Have a good day.